The OG of cancer therapy vaccines is back. But mRNA is the hot new thing. So if you're an investor, which angle do you back? I'm Rachel Williamson, and this is Phase 3. In this episode, we're getting out the tape measure and putting two Australian innovation centres back to back. At the University of Queensland, the base mRNA manufacturing facility is cashed up and going deep on cancer. In Melbourne, a team of researchers is reviving dendritic cells. They're the source of the first success in cancer therapy vaccines, and a nightmare that still haunts investors' dreams. They're both operating in a global landscape that is leaning heavily to mRNA. BioNTech, Moderna and Merck have Phase 3 trials underway for non-small cell lung cancer, and the latter two also have a vaccine for melanoma in a Phase 3 trial. And they and a gamut of researchers from the likes of the University of Florida to the Memorial Sloan Kettering Cancer Center are showing their mRNA vaccines work for a range of hard-to-treat cancers. What all of these companies and organisations are aiming for is to be the second cancer therapy ever to win US Federal Drug Administration approval. Filling the mRNA cancer gap in the Australian market is the project led by molecular biologist Dr Seth Cheetham out of Queensland, and that is BASE. The federal government funded the University of Queensland facility in 2021 to solve, at the time, a major problem. Australia had 0 mRNA manufacturing sites.
And that kind of left us exposed during the COVID-19 pandemic, because you really saw the success of the Moderna and Pfizer BioNTech COVID vaccines, which were based on the mRNA technology.
There wasn't time to build a factory and start making mRNA vaccines in 2021. But the federal government didn't want Australia to miss out.
So they set up BASE, which is a collaboration between Therapeutic Innovation Australia, which is a federal government body that funds infrastructure around Australia, and the University of Queensland. And the mission of BASE was initially very simple, which was just to provide high-quality mRNA to researchers around Australia. What's nice about that is that it's really given us that kind of view through the sector, and we've been able to see how the technology has evolved and how the applications of it have really changed over time.
Fast forward to 2023, and COVID isn't such a feature of our lives anymore. But they doubled down, and this time as a cancer vaccine hub. Seth and his team have seen a shift from COVID-19 vaccines to trying mRNA out on chronic conditions, and now a really deep swing to cancer. This year, they got a $3.3 million grant from the Medical Research Future Fund to do personalised mRNA cancer vaccines. So I had to ask, why cancer? What is it about this particular disease that is moving the dial for mRNA research funding?
I guess the reason that we applied and were successful in getting this specific funding is cancer vaccines are quite fundamentally different from other kinds of mRNA products. And the challenges here are really different from infectious disease vaccines. So infectious disease vaccines, it's all about scale. You're trying to vaccinate the entire world's population. For cancer vaccines, it's about speed. So you're really trying to turn that vaccine around as quickly as possible, but only enough for a single person.
But why cancer vaccines? Why not one of the other mRNA applications?
We've kind of settled on quite a strong internal focus on cancer. And the reason for that is, it's a very new technology, right? There's very promising results coming from early phase clinical trials from BioNTech and Moderna and some academic centers. And it's a very exciting application of the technology. So, the word personalized medicine gets thrown around a lot, but usually when people talk about personalized medicine, they're talking about using advanced diagnostics to really match a drug to a patient. So you have the best chance of that drug working for that individual. But this is an application where you're talking about actually manufacturing from scratch a drug for a single person. So it was really kind of the right time, I guess, to kind of establish this focus.
in cancer vaccines in a way that I haven't seen across other biotech fields. You have a really strong two-hander between commercial companies and organizations like yours. Normally, it's the biotechs that claim all the glory. They're running the trials, they've got the results, it's their thing. But in this particular area, you've got the LungVax clinical trial in the UK. That's University of Oxford is leading that, but that's using AstraZeneca's tech. BioNTech is using La Trobe University's new mRNA manufacturing facility. And in the US, you've got a range of clinical trials being run by the Cleveland Clinic, the Memorial Sloan Kettering Cancer Center, University of Texas. They're taking the lead on this. So why are not-for-profits, research entities, academia taking the lead in a way that they perhaps haven't in the past?
Yeah, I think part of the reason for that is that the mRNA technology, while it's been kind of worked on quite a lot in the background for a long time, there's so much innovation still happening, right? So this isn't like a completely mature, finished technology. So for example, there was a recent paper in Cell earlier this year from the University of Florida where they had a completely different way of constructing these mRNA vaccines using this kind of layered onion-like structure with lipids to deliver mRNA vaccines for brain cancer. So there's a huge amount of that kind of technical development that's still happening. The other thing I would point to as well is a lot of these trials are quite complex. So it's a really different way of doing clinical development. So in a typical clinical trial, you've got your cohort of patients and they're all getting the same thing. In this case, everyone's getting what is essentially a different drug. And so there's a lot of innovation that's still happening on the trial design side of things and also proving that technology. So that's why you see a lot of collaboration between some of the really large academic medical institutes, such as like Memorial Sloan Kettering, collaborating with BioNTech on some of their vaccines. So I think this real synergy between the biotech landscape and the universities is kind of because of the rapid emergence of this field and that there's so much innovation going on.
Seth says the cancer mRNA field is reminiscent of the emergence of biotechnology in the first place, from collaborations at a handful of Californian universities to the creation of industry pioneers such as Genentech. But figuring out how to make mRNA work for cancer is just the start of the journey. Seth mentioned the challenge of creating personalised medicine for each individual patient. But what does that even look like in massive phase 3 trials, or even once it's approved and on the market? How do you inoculate thousands of people with their own personalised vaccine, and how do you do that quickly? I asked Seth whether that was the role Base might play in a future with an mRNA cancer vaccine.
So what you really need is a very agile manufacturing solution. In addition to the cancer vaccine grant that we recently got, we also recently got funding from the MRFF to establish a phase 1 trial manufacture site, which is over at the Translational Research Institute, which is under development at the moment. And so for a traditional drug, when you're looking at getting that into patients, it's usually manufactured at a huge site. So one of the nice things about mRNA technology is that it is so scalable and it has a very small footprint compared to, for example, small molecule drugs. So you can really have these like relatively small manufacture sites that can enable clinical trials. So that's exactly what we're looking at establishing. And for these personalized vaccines, that's really key because you want the product, you don't want to deliver it at huge scale, but you want it quickly.
How fast? How fast will you be able to receive the material that you need to make a vaccine and then get it to a patient?
Yeah, so in theory, these things can be made as quickly as about 8 weeks, which is very fast. It's a very complex manufacturing solution because if you're thinking about a clinical trial that might have, even as few as 10 patients in it in an early stage trial, you're still having to manufacture 10 different drugs for that trial. So there is certainly a large amount of complexity here. But the good thing about mRNA is that you can make it really quickly and you don't need a bespoke manufacturing solution for each mRNA. So if you're thinking about manufacturing other kinds of biomolecules like proteins, each one that you manufacture will behave really differently. But with the mRNA vaccines, each one that you manufacture will behave in that manufacturing pipeline very similarly, which allows you to kind of turn them around much faster.
It helps that they're also not working with DNA extracted from bacteria in a lab. That's a source of DNA which is so problematic that a Nature article this year said nearly half of samples have design errors. Instead, they're pioneering a synthetic DNA template and cutting weeks off the manufacturing process. But Seth isn't just in charge of streamlining getting mRNA from A to B faster. His team is doing some of the new basic research into cancer vaccines themselves. I asked Seth what they're up to.
We're just getting started at the moment, but certainly the areas that we're focusing on are kind of improving the potency of the mRNA vaccines, both through engineering the mRNA itself, so looking at how different modifications can enhance that immune reaction that you get, and also different formulations. So we recently had a manuscript that we put online on using targeted delivery, so to being able to deliver the vaccine specifically to different cell types. There's a huge amount still left to be uncovered even about what an effective vaccine looks like. So, for example, the vaccines that companies like Moderna are using might have 34 different sequences that they're trying to raise immunity against. But in a patient, when they inject that, it might only be one or two that's actually driving immunity. So there's a huge amount of work to be done there in terms of understanding why particular antigens are effective and others aren't. So I would say the field of mRNA cancer vaccines is very embryonic, and there's a huge amount of potential for these things to get even better than they are now.
That was BASE Group Leader and Deputy Director Dr Seth Cheetham. Seth's backers are convinced mRNA cancer vaccines are on track to become a real therapy. They're basing their view on positive results from global clinical trials, the multi-sector collaborations, and the support this concept is getting from other countries and funders. But could there be something even better out there? Something that has been proved to work before? We'll find out after the break.
Hi there, I'm Charis Palmer, producer of Phase 3. When Rachel and I set about building a new podcast for life science leaders, scientists and long-suffering biotech investors, we looked at what was missing in this space. We believe Phase 3 serves an unmet need for in-depth conversations in a world where nuance matters and AI-written investment articles simply won't cut it. If you agree, please follow us and sign up to our newsletter via LinkedIn. Pledge financial support at phase3.buzzsprout.com and rate and review the podcast on the podcast platform you use to help bring it to the attention of others. Now back to the show.
If mRNA is the cancer vaccine therapy of the future, then dendritic cells are the vaccines of the past. Dendritic cells were discovered by Canadian physician Dr Ralph Steinman in 1973. In 2011, he won the Nobel Prize in Physiology or Medicine for his discovery, 3 days after he died. Normally you have to be alive to win a Nobel, but the committee decided that since he wasn't dead when the decision was made, he still qualified. For many years, researchers have been trying to get DCs, as they're known, to work for cancer immunotherapy. That's because DCs are the cells that patrol the body and find foreign invaders. They then educate the soldier cells, the T cells, to attack. The general idea in the past was to take the DCs out of a patient, rub their little noses in a cancer antigen, and put them back in. And in the 1990s and 2000s, there was a huge number of clinical trials using monocyte-derived dendritic cells to try and do this. Of all of these trials and all of the money that backed them, just one drug, Provenge, for late-stage prostate cancer, was approved. That was 2010. It was so expensive it bankrupted its owner Dendreon. It was revived at the end of the decade to treat early-stage prostate cancer. But in many ways, the 1990s and 2000s were 2 decades wasted. And this is why.
It turns out they're the wrong ones. We've been using the wrong DCs for a long while, in my opinion.
This is Shalin Naik. He's the lab head at the Walter and Eliza Hall Institute of Medical Research, or WEHI for short. His research is the foundation for a potential revival of dendritic cell cancer therapy vaccines.
We didn't know any better until Ken Shortman at WEHI in 1992 discovered this obscure subset of dendritic cells called the DC1s. He called them the CD8 positive dendritic cells. Anyway, it turns out this is the crucial subtype for kickstarting cytotoxic T cell immunity, the killer immune cells. But no one's been able to make them in large numbers in a dish, and that's because they're super tricky.
Why are they so tricky?
So we've been chasing the wrong growth hormones to grow DC1s. We knew the ones that grew monocyte-derived DCs, not the one that grew DC1s. Then during my PhD with Ken Shortman back in the early 2000s, I discovered that it was FLT3 ligand that in culture could really stimulate great DC1 production in mouse. So then I sat back, I published the paper in 2005, I sat back, I said, right, someone's going to go and make these in a human form and inject them into patients, and then here we go. And I was waiting, and no one did.
Had people just been too burned by trying to make another Gardasil, trying to make DC vaccines work, and just were like, we're not?
Yeah, I reckon that's part of it. People still are. Investors are seriously burnt by monocyte-derived DCs, so they're like gun shy about any use of the word dendritic cell. They kind of run a mile, but it's gradually creeping back into the kind of collective psyche that this is something we need to be thinking about.
Shalin and his team have spent the last few years figuring out how to grow DC1s from human stem cells. They've found a secret sauce that can make 10 times more DC1s at 10 times greater purity. So they've solved two problems: finding a dendritic cell that will work and getting enough to test it on a real live human.
It suddenly tips the balance that we can inject a decent number of DCs that would be comparable to what we've done in the monocyte-derived DC area to really test the idea. Can we test this? Does it work? And if you can't generate the numbers, you can't test if it works. So we finally cracked that problem.
Shalin, one of the problems with the original attempts at DC vaccines was they could only get an immune response of 5 to 15%. And this is because the way solid tumours suppress immune activity also dampens the behaviour of dendritic cells. So what are the other unsolved challenges that you're having to wade through now? And with peptide and mRNA vaccines getting so close, what's your competitive advantage with DC1s?
Yeah, those are two really great questions. The first question is, just because you can make enough of them, will they go and work in the tumour? And the answer is, we hope so, but possibly not. And the reason is that we know that the tumour microenvironment is highly immunosuppressive, and that is also the case for dendritic cells. So cytokines like IL-10, TGF-beta, basically the tumour secretes these factors that dampen down the DCs, they kind of turn them off, and they can't do their job. But the great thing is we're partnering with a lab here next to us at WEHI from Stephen Nutt's group. And Stephen Nutt's group have developed a novel CAR that is dendritic cell specific. So a reminder, chimeric antigen receptors, which is CAR T cells, they've revolutionized the immunotherapy of hematological cancers, liquid cancers like lymphoma and myeloma now. But they haven't really worked in solid cancers. But what the Nutt lab have done has taken a CAR that binds a cancer, changed the cytoplasmic tail to be very DC specific to activate them, and now that solves what in the dendritic cell we call the DC activation timing problem.
Shalin says you've got to time DC activation perfectly. If it's too early, the DCs won't see the tumour antigen. If it's too late, it could start working for the enemy. The tumour can actually put it to work suppressing the immune response to the cancer. He says it means there are a few moving parts they need to think about.
We're really thinking about how do we get it into the tumour, how do we make sure it's not immunosuppressed, and how do we kickstart the immune response in the appropriate way once we do see the cancer.
And so how are dendritic cell vaccines, and specifically DC1s, going to have an advantage over other cancer vaccine theories?
So we think it just comes down to two words: epitope spreading.
Great. What does that mean?
What does epitope spreading mean? All right. Epitopes are the cancer antigens that are different from normal tissue that allow the immune response to recognize it and kill it. But the tumor evolves and it changes, and there's these new antigens arise, but also if you go and attack those tumor cells, then some of the tumor cells that mutated those further or didn't have them might rise again. So this idea of tumor heterogeneity means it can be like a Medusa's head. It can just grow out by avoiding several arms of the immune response. And so while we believe that peptide vaccines, personalized antigen vaccines could be useful, they're also liable to escape because you have to make a choice. And if you choose, 7 antigens, but there's a plethora of antigens, if you get those wrong or they don't eradicate 100% of the cancer, a cancer can bounce back and you have to go back to the drawing board. The beauty of dendritic cells is it is agnostic to the antigen. It means it doesn't care. It's like whatever is there, it will take up, it'll process, and it'll present to the T cell. And if there is a T cell around that recognizes that, it will get activated. So what epitope spreading is, the DC takes up the tumor, it activates the T cells. The T cells go and kill the tumor cells that have that antigen. If some of them escape, new DCs can pick up new tumors, educate new T cells for the new antigens, go back and they will get killed, and so on and so forth. So once you kickstart that process, it's sort of like a self-sustaining therapy. And that's how checkpoint inhibitors work. It's not only the T cells that are happening here. It's the whole network of dendritic cells, T cells, tumor killing, new antigens allyzed, and so on. And so we believe dendritic cell therapy will work similarly, but in an advanced way compared to checkpoint inhibitors, in that it will allow epitope spreading and a tumor clearance longer term. That's the hypothesis.
Shalin is hoping human trials at the Peter MacCallum Cancer Centre could be just a couple of years away. Which raises an interesting question. He works at the purer end of medical research. So I asked him, how much do researchers at this end of the biotech spectrum have to factor in crass commercial considerations? Like producing something that is simple to make and not eye-wateringly expensive over something that just works.
So I'm new to this world, right? I'm very much a dendritic cell biology researcher. I am not a clinician. I am not a biotech company CEO.
So you're the perfect person to be asking about this then, because this is something that maybe you are thinking about right now.
There's certainly considerations there. But the other advice we have is get it into patients, see if it works. And you don't have to be perfect in the first trial for cost in that regard. It will be expensive. Cell therapies are expensive by definition. But if it works, well, then we can think about how do we make the manufacturing process shorter? How do we make it cheaper? First up, we'll be doing autologous dendritic cells, which basically means the patient's own DCs. But in the future, can we have an off-the-shelf product? Can we take cord blood stem cells from newborn babies? They're a renewable resource as long as we keep having them. Have a DC1 biobank. And when a patient comes along, we just do a matching exercise like we do with bone marrow transplants. We match them, we pull them out of the freezer, and we treat the patient. That's the ultimate goal. Now, whether it's from a baby-derived stem cell or an iPSC-derived DC1, that remains to be seen.
mRNA vaccines have kind of been the reason why cancer vaccines have been popularized, particularly this year. How do you think you guys are going to go up against that concept, which has so many big names behind it, has so much money behind it? You're coming at it from academia and going, this works, we can do this, we can make it work, versus Merck and Pfizer being like, we've just spent a huge amount of money on huge mRNA manufacturing facilities, and why would we even look at that when we're spending so much money elsewhere?
Yeah, I completely agree that is something we should be thinking about. So mRNA is great. It's fantastic. In fact, we use mRNA LNPs in our manufacturing process. They're a fantastic delivery modality, either in a dish or directly in a patient. They've been tested. They're by and large safe. They are efficacious at generating an immune response and the right kind of immune response. Is it going to work for cancer? You have to choose something to target. So is that choice a pan-cancer vaccine? I think it's going to be unlikely you find a magic bullet there. Is it going to be a personalized vaccine? That's the kind of approach many people are taking. You sequence the tumor, you identify the cancer-specific antigens, you formulate it in a mRNA and you deliver it into a patient. But again, if tumor heterogeneity is sufficient to bypass those few peptides that you select in the vaccine, well, then you're back at square one.
That was WEHI laboratory head Dr Shalin Naik. Shalin found the way to make lots of the kinds of dendritic cells that actually do what we want them to in a cancer vaccine. But his current work is a long way back in the race to build a successful and monetised cancer therapy vaccine. In our next and final episode of VAXXED, we ask our usual question: what does the future hold for this emerging line of cancer therapy inquiry? How does it fit into the competitive constellation of cancer cures? And which modality will be the first to commercialisation?